Rahim. Assalamu alaikum and good day, everybody. I'm Hazima Mozam and I welcome you to 21st Comstech Distinguished Scholars webinar series. We are very delighted and honored to have Professor Dr. Michael Threadgill with us today. Professor Dr. Michael Threadgill is an eminent chemist, and to introduce him briefly, he is Fellow of Royal Society of Chemistry. He's Professor Emeritus at University of Bath. He obtained his PhD from University of Cambridge. To his credit, he has 160 full papers and 5 patents to his name. He has supervised 35 PhD students. And with this, today, the title of his talk is Structure-Based Design and Development of Inhibitors of Tankyrases, Drug Targets in Cancer and Diabetes. And with this, I would request Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS, to formally welcome our guest speaker today.
It's such a delight to welcome Mike. Mike is a personal friend. He is not only a very good scientist, but also a wonderful human being, so deeply connected to the rest of the world. So I consider him as part of the global humanity, someone of global vision, and he has been helping people from all over the developing world. His affiliation with Pakistan is very strong. We have been working together with him since long. We have had the honor of welcoming Mike multiple times, and every time it was an immense learning experience. So Mike's human attributes are well above his credentials, which has been presented. The fact is that science becomes effective when it is done by a person of good heart. It is very important because science with intention, science with the sincerity of purpose, science for spreading knowledge is exactly what he does. And this is where we feel Mike is different from the other. So it is indeed a great pleasure for us to have Professor Dr. Mike Threadgill as Comstech Distinguished Speaker. Mike, for just a brief introduction of Comstech. Comstech is a very big organization in OIC, which is Organization of Islamic Countries. So it is like a UN system. This is the second largest group of nations under the OIC banner. And Science and Technology Standing Committee is located in Islamabad, just in front of the Prime Minister's Secretariat. I was given the responsibility of leading this institution. So the benefit of our existing linkages at the ICCBS are now spread to far and wide. And you would be pleased to know that hall is already full with over 100 people from some 13 countries, as I can see. With this, I would like to request Professor Dr. Mike Threadgill for his exciting lecture on a very, very important topic.
Well, thank you very much for that kind introduction, Professor Iqbal. Our friendship goes back many years, and I'm very grateful for it. And I've also in the past been able to welcome some of your students into my lab for visits of a few months. And going forward, Dr. Hina has kindly shared one of her students with me as an external supervisor. I'm looking forward to that. I'm really looking forward to that. So on with the lecture. Some of you who have been to meetings in Karachi may have seen some of this work before. So if you fall asleep during those parts, I won't worry too much. So on with the lecture. Let's share the screen. There we go. So my lecture title is going to cover some of the work that we've done in my lab at Bath, probably over the last 10 years on inhibitors of tankerase. But the story goes back beyond that. The story goes back beyond that because I've been working on the whole family of poly-ADP ribose polymerases for, gosh, it's nearly 30 years now. But a little bit about me in the context. I was for many years a researcher and academic at the University of Bath. Down here we can see in England. I retired nearly 3 years ago, well, officially retired, and moved house to Cardiff, which is the capital of Wales. And I now do some part-time research and teaching work with Aberystwyth University in central Wales. But so this talk is going to cover some work from my time at Bath. And the tankyrases project I'm not involved with anymore. This is in a sense a historical talk up to the time I left Bath, up to the time I retired from Bath. But the research work on tankyrases from my group is being carried on by my colleague, Dr. Amit Nathubai, who is now at the University of Sunderland. So enough of the introduction. Let's get on with the science. And I'm having trouble clicking forward. There we go. So as I said, we've been working for about 30 years on different members of the family of poly-ADP ribose polymerases. When I started, there was only one isoform known, which we now know as PARP1. And this has been very well researched by a large number of groups. It's involved in all sorts of cellular processes, and inhibitors are already in clinical use for the treatment of cancer. Although we and others have shown that there's potential for these PARP1 inhibitors in other diseases as well, in ischemia reperfusion diseases and in hemorrhagic shock. And I'm rather hoping that these inhibitors will move out of the cancer, spread out of the cancer area into treatment of these other disorders. There are 17 isoforms, or 17 members of this superfamily. PARP2 has a similar function. PARP3 we've also worked on. So this slide shows the family members that we have done some work on. PARP1, PARP2, PARP3, done a little bit of work on PARP3. PARP14, did a little bit of work on PARP14 just before I retired. But the work I want to tell you about is the work on PARP5A, PARP5B, which are known as tankyrase 1 and tankyrase 2. Two very similar proteins, particularly around the catalytic site. I'll show you in the next slide what the catalytic site does, but two very similar proteins in terms of their function and in terms of their structure at the catalytic site. Other sites remote from the catalytic site differ between the two tankyrases. So tankyrase 1 has roles in regulating the length of the telomeres. It has roles in the function of the mitotic spindle and in the Wnt proliferation signaling system, where it carries out a post-translational modification on axin. Axin is a component of the axin beta-catenin system, where beta-catenin, if it's present in too high a concentration in the cell, will translocate to the nucleus and switch on genes for proliferation. So we can see here that in the context of cancer, inhibiting the growth of telomeres, inhibiting the function of the mitotic spindle, and inhibiting Wnt proliferation signaling through inhibiting poly-ADP ribosylation of axin will be a good thing in terms of treating cancer. But tankyrases also have other functions in the cell. One of those is in regulating glucose uptake into adipose tissue and other cells, other peripheral cells, where its target is IRAP. So tankerase 1 is a drug target in cancer and in diabetes. Tankerase 2 has been less well studied, but it's certainly involved in the mitotic spindle, involved in the Wnt signaling, where it again poly-ADP ribosylates the target axin, and it's involved in glucose uptake. So we think that's a drug target in cancer and diabetes. And when we set out to look at this on this program, we didn't seek to distinguish between tankerase 1 and tankerase 2. We thought that inhibition of both of them would be good. So we were not looking for selectivity for tankerase 1 over tankerase 2, but we did want to design compounds which inhibited the tankerases and not other members of the PARP family, and also not other enzymes that use NAD. Was there about 100 enzymes, maybe more than 100 enzymes in the human cell which use NAD? We do not want to get off-target toxicity by inhibiting these other enzymes. So you see some selectivity studies as we go through. So a quick cartoon of the telomere. For those of us who are chemists not necessarily that familiar with the function of DNA and in the replication of DNA, I've represented a chromosome as just a simple bar. It's much more complicated than that. But the genes and introns are located in the chromosome, but each chromosome is capped with a piece of what was thought originally to be junk DNA, non-coding DNA at the ends. And this was called the telomere. But this telomere actually has a function. It consists of double-stranded DNA, which doubles back on itself, And this is important because it stops the cell recognizing the ends of the chromosome as a broken end. The end is wrapped in on itself, so that the DNA repair systems don't recognize the end of the chromosome, the natural end of the chromosome, as a double-stranded break. So the genomic DNA is here. This is the telomere with a loop structure. You also see a protein here called TRF1. We'll see that on a later slide. In fact, here it is, TRF1. I've now represented it in a linear way, just as a cartoon. So TRF1 binds to the telomere. Tankerase 1 takes the ADP ribosome unit off NAD, liberating nicotinamide, and generates a polymer of ADP ribose attached to the TRF1. This makes the TRF1 lift off from the telomere. So another complex of enzymes called telomerase can bind to the telomere and extend the length of the telomere. Why is this important? It's important because at each replication cycle of a cell, which is obviously what's going on in cancer, at each replication of a cell, the DNA is copied, but not right up to the end. So the telomeres actually get shorter as at each replication cycle. The telomeres get shorter. And if the telomere wasn't re-lengthened in each cycle, then eventually the telomeres would disappear and the cell would be in a crisis, in that in the next replication cycle, some of the genomic material would not be copied. So this is a replication crisis that a cell would have. So tankerase, if you like, regulates the length of the telomere by removing this inhibitory protein TRF1 to allow telomerase to re-lengthen the telomere. And the logic is that if we inhibit tankerase 1, then we'll stop the telomeres re-lengthening and push the cancer cells into a crisis. as they replicate. I do have other cartoons covering the Wnt pathway and NUMA and the mitotic spindle, but I want to get on to what we've done rather than the background. So this is just one illustration of why inhibiting a tankerase is a good thing in terms of the treatment of cancer. So what do tankerases actually do in mechanistic terms? Most of us are chemists, I think, in the audience. I hope so. The tankerases do what all the poly-ADP ribose polymerases do. They use NAD as a substrate. This is NAD, and they bind through 3 very strong hydrogen bonds onto the amide part of the nicotinamide. And this actually stretches this carbon-nitrogen bond, stretches the carbon-nitrogen bond, and it breaks to generate this intermediate cation. And the leaving group is ADP. Sorry, sorry, I'll go back from there. This cation is captured by an oxygen nucleophile, which could be an oxygen nucleophile on the protein that's being modified, or it could be a ribose oxygen nucleophile in the growing polymer. So nicotinamide is the leaving group, and its leaving group ability is enhanced by these 3 strong hydrogen bonds. And so the nucleophile becomes attached to the ADP ribose. So here's one ADP ribose unit joined on, and you can see that the enzyme builds up a polymer of ADP ribose units. And this is a post-translational modification of the enzyme, which regulates the function of the enzyme. In the case of TRF1, it causes it to lift off from the telomere. The tankerases produce polymers of about 30 to 50 units in length. PARP1 produces much larger polymers of 200, 300, 400 units in length. So they're quite long polymers. So how did we start? About a bit under 10 years ago, how did we start? Well, we've been working on PARP1 for several years before that, and we've got some inhibitors. And 3 inhibitors here had about the same activity against PARP1. 5-AIQ, which has become one of the standard inhibitors, actually came from Warner Lambert Park Davis, but we developed it much further. And these 2 analogues. The activity against PARP1 is about the same. We noticed that these 2 compounds weren't really very toxic, cytotoxic towards 2 different human cancer cell lines. This is really not very potent. 100 micromolar is really not active. But what we did notice is that these compounds are also inactive against tankerase 1. We had a very crude assay at that time for tankerase 1. And they were also inactive against tankerase 1. Well, we did notice this compound with the phenyl here in the 3 position of the isoquinolinone was cytotoxic towards these two human tumor cell lines and showed some activity, some good activity, about 100 nanomolar against tankerase 1. That's one start point for our study. Gives us a hint that this kind of structure with a benzene ring here is probably a good bet, a good start point for designing inhibitors. But that's looking at the inhibitors. That was a chance discovery. We just noticed that these compounds with benzene rings here in this position were particularly cytotoxic. So the cytotoxicity was not being mediated through inhibition of PARP1. That shouldn't be a cytotoxic event, but it was being mediated through something else. And there was a correlation here not necessarily a causation, but a correlation. What was the other start point? The other start point was really from some crystal structures in collaboration with our colleague Larry Lehtiö in Finland. This compound, even though 5-AIQ, even though it doesn't inhibit tankerase 1 particularly well, it will bind. It will bind. And Larry soaked this compound into crystals, actually of tankerase 2. Tankerase 2 happens to be easier to crystallize, and obtained some crystal structures. We noticed that it makes the 3 hydrogen bonds, which are the critical ones for the hydrogen bonding of the natural substrate, nicotinamide. So it's this motif at the top here, this secondary amide, which seems to be very important in binding into the enzyme. This is another representation of it, just with space filling, showing that it's the serine and the glycine. It fits very snugly into the nicotinamide binding site on the tankerase 2, very snugly. And if we draw a cartoon of that, if we draw just a rough cartoon, we can see that this is binding in the nicotinamide binding site. We can see it buried in there. This is the surface of the enzyme. But we can see in front of this, there's a cavity, which is actually lined by hydrophobic residues. And we're peering down a tunnel from the outside into this cavity through to the nicotinamide binding site. When we started, just as we started, there was published the first inhibitor of the tankerases, which was claimed to be selective. And I will say claimed at this point, because we've since found it not to be as selective as the original authors claimed. XAV939, with a similar motif in this region, secondary amide as part of a lactam, but with a benzene ring and CF3 pointing in this direction. And this benzene ring and CF3 will occupy this hydrophobic cavity. Actually, this CF3 just starts to poke into this narrower tunnel through to the outside. So what did we do? We're chemists. We started making lots of compounds, but we wanted to answer a number of these questions. Around this core structure, does this ring have to be aromatic or can it be saturated? XAV939 was saturated. Our lead compound, or our first hit, was aromatic. Does this have to be aromatic? Bulky, shape? What about substitution? Electron withdrawing groups, electron donating groups, polar, non-polar, all the things that medicinal chemists think about. We thought it was essential to keep this part in the grey box. Substitution in this position, electron withdrawing, electron donating, and so on. And what should go in this position? The first set of compounds that we made were quinazolinones, with an aromatic group in the 2 position. And to start with, we varied the substituent Y in the para position and kept R constant as methyl. What I'm going to do is show you just a snapshot of a few of the compounds. We made many, many more, but just to illustrate some structure-activity relationship points. Varying Y doesn't seem to make much difference to the inhibition of the tankerases. See, we're in units or tens of nanomolar here. So we have plenty of activity. In terms of selectivity, if the Y group is hydrophobic, then we have selectivity. We have poor activity against part 1 and part 2. We initially tested the compounds against an oxidoreductase that uses NAD as well, IMPDH, and found them essentially inactive. So early parts of the study, we were counter-screening against IMPDH. We stopped doing that because these compounds just weren't inhibiting IMPDH at all. What we did notice was that when the para substituent was polar hydrogen bond donor, we started to get PARP1 inhibitory activity. So we lose selectivity, retain potency, lose selectivity. Extending that to a charged functionality, a CH2NH3+, trying to get some water solubility, the tankerase activity drops off, but the PARP activity increases. So this is really not a useful compound. We tested this compound with the CBZ protection still on it, really because we test all our intermediates as well. And it retains some activity. Even this is an enormous group, it retains some activity. Because it's relatively non-polar, we get selectivity against, so it doesn't inhibit PARP1, PARP2. It still retains good activity against tankyrases. It even gets some activity when we put a big bulky ferrocene in that position, but it does drop off quite a lot. The final set I want to show here is when we're varying this group R, but keeping the para group Y constant. So with hydrogen at that position, activity drops off, methoxy activity drops off from the initial methyl. With hydroxy, hydrogen bonding donor, we still have good activity, but we're getting PARP1 inhibitory activity as well. Same story as in position Y as position R. Introduce a hydrogen bond donor, a polar hydrogen bond donor, and we lose selectivity. We retain activity, but lose selectivity. So polar in both of those regions is not a good plan. We made a lot more quinazoline, but here I'm going to show briefly some work we did on the isoquinoline. So we've got CH here rather than nitrogen. We asked the similar sort of questions. For the keen synthetic chemists amongst you, here's the way we made them. Here's the way we made one set with amino in the 5 position here, and a large number of groups, different functionalities in this position. We'll see those in a minute. The key step was a Suzuki reaction, so we could introduce diversity fairly late in the synthesis. And we had the usual set of substituents here to test different features. To put different R groups in here, in at the 5 position, we used this chemistry to see how we could access compounds with fluorine, methyl, and methoxy. Large number of different substituents in this region. And in the isoquinoline series, it was actually quite easy to introduce compounds substituted at the meta position and the ortho position, as well as the para position. And from the methoxy compound, we could access the hydroxy compounds. And here are some of the more weird and wonderful compounds that we also made, rather than just simple derivatives. We put a pyridine in, this bicycle to increase a bit of bulk, put a methyl in at this position. That's going to be important, we'll see soon, because that modulates the conformation of the molecule, the conformation of this benzene ring relative to this bicycle. We also put some basic heterocycles in this position. We'd had trouble with the NH3+ compound, which we saw before in the quinazoline series, which was essentially inactive. But we thought if we introduced some more lipophilicity here, we might get water solubility without losing activity. And really then, because the PhD student, Helen Payne, fancied making a long, thin substituent, we put this very long thin substituent on in the 3 position of the isoquinolinone. We didn't really expect this to be active. This was just an exercise in, if you like, fun chemistry. So different properties of different side chains. Again, Helen made a lot of compounds in this series, but I'm just going to use some to illustrate some structure activity features. First of all, let's look at keeping Y constant, but R is varied. Amino in that position, hydrogen bond donor, good activity, but selectivity is not brilliant. The selectivity is not brilliant. And that fits in with the quinazoline work. Methyl is good, and notice now we're getting down to units of nanomolar against tankerase 2. Part 1 and part 2 essentially inactive. Fluorine and methoxy, similar sort of story. Hydrogen bond donor is a really bad idea in terms of activity, but not necessarily in terms of potency at position R. Now just picking up on this ring in blue, we put chlorine on in different positions. The parachloro, good activity and good selectivity. Moving the chlorine round to the meta position, we lose some activity, but it's still adequate. Move it into the 2 position, we severely lost activity. We had no activity against tankerase 1 and greater than 100 nanomolar IC50 against tankerase 2. Putting 2 ortho substituents in really made things much worse. So that's telling us something about the requirements in that ring. Best is a parasubstituent. Meta-substituent is okay. Ortho-substituents are bad. Bulky ortho-substituents like chlorine. In a couple of slides' time, I'll come on to show you why that is. Now we're comparing these different groups in this position. So we have that long thin one, putting that long thin substituent on kills the activity against tankerase 1, but we still have good activity against tankerase 2. Accidentally, we discovered an isoform selective inhibitor. We don't really want one, but because we'd like to inhibit both isoforms. CH2 piperidine at the Y position, at Y para position, good activity. This is also a water-soluble compound, as its hydrochloride salt. The pyridinyl, this one, good activity, but loss of selectivity. This is that compound is actually quite a good part 1 inhibitor. It's almost as good as some of the ones on the market. And notice that when we put a methyl in this red position here, we retain activity. So putting steric bulk in these 2 ortho positions here with a chlorine destroys activity, but putting a methyl group in here reduces activity, but it's not bad. It's not bad. We thought we had to explain that. Why a group here is acceptable, but not optimal. Why groups here and here are not. So we're just comparing these compounds with this compound. And we looked at the conformation of the molecules to try to explain that. So here we have the dichloro, poor activity. The compound with the methyl here, where there should be a steric clash across here, good activity. Steric clashes here and here. And we're going to compare them with this compound, which is probably the best one in the isoquinolinone series. Let's have a look at the structures. If we do a small molecule crystal structure on this dichloro compound, we notice that the dihedral angle here is 67 degrees. So this benzene ring is twisted out of plane with this bicycle by 67 degrees. But here in this best hit, or similar to the best hit, without any substituents in this region, the dihedral angle is only 7 degrees. Is that the issue? Is that what's going on? Is that the problem? Is it that this benzene ring is being twisted out of the general plane of the isoquinolinone? Is that the problem? We couldn't actually get a small molecule crystal structure of the methyl compound, but let's look at how these compounds actually bind into the enzyme. Does this small molecule twist, dihedral twist, translate into the binding into the enzyme? Even though this is a poor inhibitor, we can still bind it and get a crystal structure. We measure the dihedral angle as being 60 degrees here again. It's actually twisted in the opposite sense to the small molecule crystal structure, but that's not important. Here, the dihedral angle is much smaller when we haven't got any ortho substituents here, here, or here. So in plane seems to be active, out of plane seems to be inactive. Let's look at this compound though, where we would predict a steric clash across here if this was in the plane of the isoquinolinone. As I said, we couldn't get a small molecule crystal structure, but we've got a crystal structure in the enzyme. The dihedral angle is 50 degrees. Over here with this methoxy compound, no substituents here. Dihedral angle is 8 degrees. Large dihedral angle, small dihedral angle, but both active. Both active. So it's not this twist. Our hypothesis that it was this dihedral twist that was causing the inactivity was not true. We showed that that hypothesis was false. It's actually the presence of the big ortho substituents here and here in the dichloro compound. And when we modeled it, we found that they were actually bumping into, colliding with the surface of the enzyme at the entrance to the hydrophobic pocket. So here we go. Substituent in this position is okay. Twisting this benzene ring out of plane is actually okay. When we looked further into the hydrophobic pocket, we found that actually the hydrophobic pocket is almost spherical. The hole in the enzyme is almost spherical. So it will take any sort of twist here. And it's about the size of a benzene ring twisted through 360 degrees. You can just rotate it freely in that space. Okay. That is a very quick run through of a lot of work by our collaborators in terms of the crystallography, by Helen Payne, a PhD student, now a part-time academic at the University of Bath, and by my colleague, postdoc at the time, Amit Nathubai, who did all the work on the quinazolinones. I'm now going to move into addressing another question. Does this ring have to be aromatic? And can we put substituents on it? So we made compounds of these general types. We put nitrogen around the ring here, and in this compound, naphthyridinones, and these are analogues with nitrogen in the quinazolone type series. We went saturated as well. Nitrogen here, nitrogen here. Compounds with nitrogen here were actually more difficult to synthesize, so we didn't get very far in that one. We put oxygen here, CH2, all to compare with XAV939, which was the lead compound from Novartis, a compound which was claimed to be highly potent and selective. We wanted to see if we could do the usual medicinal chemist replacement. Can we replace sulfur with oxygen, CH2? Can we put a nitrogen in with or without a substituent? We tried all of those. And this work was done by my PhD student, Katerina Kumpan from Moscow. She joined my lab at the same time as Helen Payne and did very good work in this heterocyclic chemistry. I won't go through the chemistry. That would take many slides. This work is published, so and the references at the bottom of the slides. So if you actually want to read about the work in the chemistry in detail, you can. And I'll be happy to engage in one-to-one email discussions or similar if anybody wants to know details of the chemistry. Okay, let's look at the biological activity. Here's the lead compound at the time, one of the lead compounds, the isoquinoline known. So just for reference, we put a nitrogen in and oh dear. Put a nitrogen in this position, put a nitrogen in this position, oh dear. Activity almost disappears. It drops off by over 100-fold or up to 100-fold. Maybe a little bit of activity for this compound. We quaternized this to make it positively charged, and that made life even worse. Although we did get a water-soluble compound, it didn't actually inhibit either of the tankyrases. We also made the N-oxide, which improved the activity a bit, but still not to a useful level. At that point, I was getting worried in this series, and Katerina Kumpan, the PhD student, was getting seriously worried that she may not have active compounds for her PhD thesis. We can explain this actually in terms of crystal structure. Well, explain it. We have a suggestion for the explanation. There happens to be a positively charged lysine mounted just above this aromatic ring. So when we have an electron donating ring, benzene ring, electron-rich benzene ring at this position, that's not an unfavorable interaction. It's quite a long distance for an interaction, but it's not unfavorable. When we have a cation or electron deficient ring at this position, we have cation to cation repulsion. And these are crystal structures. Even though the activity was poor, you could still get crystal structures by soaking the enzyme. This is by no means proven. I'm just floating the idea with you at the moment as to why electron deficiency in this area is not a good thing in terms of binding to tankyrases. Let's move on into saturating that ring. Here's the standard, if you like. We made this N-benzyl compound up here, inactive, took that, that was a synthetic intermediate, took the benzyl off, inactive. Here's XAV939 showing reasonable activity, but not as good selectivity as the Novartis authors claimed. And we've since shown that in cellular systems, it's almost non-selective between part 1 and tankerase 1. We got some activity, although much reduced, by replacing sulfur with CH2. We also replaced it with oxygen. That destroys activity. So positive charge in this region of space, bad. Substituent in this region of space, bad for activity. CH2 is okay, but much worse, but sulfur is the best. But the best one in this series came out of these N compounds with saturated in this ring, but N-methyl. But it was critical on the nature of the group Y. Y equals hydrogen, poor activity and poor selectivity. Turned Y, put a substituent in at Y, and activity improves enormously and so does the selectivity. So these were an interesting set of compounds. I'm not going to discuss them in this lecture, but again, we have a strong hypothesis backed up by some modeling studies as to why some saturated compounds of this nature are active and some are not. And it's all to do with how that six-membered ring puckers. It can pucker with a left-handed twist or a right-handed twist. One is good, the other is bad. So let's move forward. Let's move forward from the hydrophobic cavity, which we've filled with benzene ring. And this is XAV. Now, this is our compound with the CF3 at this position. And we notice that just starts to enter the tunnel to the exterior. We can see it here in this more open structure. This comes from a crystal structure. There's a blow up of it. We can see that CF3 is just entering that tube, that tunnel to the exterior. This is the compound I mentioned earlier. We're still with this CBZ protecting group, benzyl oxycarbonyl protecting group, when we're trying to make the NH3 plus compound. And we had activity with this, which surprised us. And when we got a crystal structure, we found that this benzene ring, this benzyl group with the benzene ring, actually protruded right through the tunnel and bound to the exterior of the enzyme. Could we exploit that? Could we actually design compounds that do that deliberately? This was a serendipitous discovery, accidental discovery. Happy accident, as they say. Well, we did some chemistry in the quinazolinone series and deliberately designed compounds with a quinazolinone here to go into the nicotinamide binding site and a quinoline at this position on the end of a linker to bind at the exterior. We thought that maybe this part would mimic the nicotinamide, and this part, this quinoline might actually mimic the adenosine of NAD. So we might get a dual site binder out of this. And we do. We do. Gain some comparative data. Here's the isoquinolinone. At that time, the best compound we have, XAV939. Our dual site inhibitor. We had one with a methyl, one without a methyl at this position. I'm showing you the data for the one with a methyl. We're now down to 100 picomolar with good selectivity. A good selectivity against part 1 and part 2. So we have now a very potent, very selective compound. We also tested the two fragments to see if they whether the benefit in terms of activity resided in one or the other. So we took the quinazolinone fragment, which was inactive because this polar carboxylic acid or carboxylate group would have to go into that hydrophobic cavity, and that's really a bad thing. It was actually a reasonable PARP inhibitor, but not a tankerase inhibitor. And the same applies with the quinoline fragment over here. No activity at all, really. No activity at all. So linking them together, we have what we think is a dual site inhibitor binding at two sites on the enzyme. Of course, we've got crystal structures. Here's the structure we saw with this benzyl oxycarbonyl compound, where the aromatic ring was bound over here to the left. Here's a similar view for our dual site inhibitor. It happens to be the one without the methyl, but the quinoline is bound in different position. It's not down here, it's up here. When we blow that up, we find it's pi stacked onto histidine 1048. And this pocket here is the pocket on the enzyme which binds to the adenosine of NAD. So we genuinely have got a dual site inhibitor. We've got part which binds into the nicotinamide binding site, and part here, which is binding into the adenosine binding site. Just to take another view, tilted it a bit so we can see the pi stack. Here's the drug, the quinoline here, stacked beautifully onto the histidine imidazole ring. So what about activity? Here we've got both compounds, 100 picomolar against tankerase 2, a bit less active against tankerase 1, but still quite good. We showed that it has some activity against Wnt signaling in the cell. This is how Novartis discovered the XAV939 compound. It wasn't designed. It was noticed that it inhibited the Wnt response in cells. And using the same assay, we've shown that we're 8 to 10 times more potent in terms of inhibiting the Wnt response. And we have good, much better selectivity against PARP1 than there is for XAV939. We have anti-proliferative activity. This is published work, and in view of the time, I won't discuss it in detail. But using a clonogenic assay, we can see that no inhibitor, 100 nanomolar and 1 micromolar against these cells, these tumor cells. We have good anti-proliferative activity. We even have some anti-proliferative activity against at 100 nanomolar in this cell type. So we're showing these are human colon carcinoma cells, cell line, where we know that that Wnt is important in driving the proliferative response. So we've got activity in that sense, cellular activity. I mentioned right at the beginning that tankerases are also involved in the uptake of the insulin-stimulated uptake of glucose into adipocytes. And here are some data that show activity of XAV939 and of our dual site inhibitors, showing that XAV is relatively inactive in this model because it's just not potent enough. Our super potent inhibitors cause increase in uptake of glucose into adipocytes, but only when they're stimulated with insulin. And this is part of the function, part of what insulin does in healthy adults, and part of the loss of function in diabetic patients, particularly in type 2 diabetic patients. We have some more data, which I can't show you, which shows the activity of both of these compounds, particularly with R equals methyl, in influencing differentiation of cells of pancreatic cells. And so we can use this to select for differentiation into insulin producing cells. So we've got another angle at using these compounds potentially in the treatment of diabetes. So these compounds are looking interesting. But as I said, I'm not active on this anymore. The leadership of this program has moved to my colleague Amit Nathubai at Sunderland. I've changed direction completely now. I still do chemistry, but not in the lab. I supervise chemistry, co-supervise chemistry students, one in Karachi and also in Aberystwyth. But in Aberystwyth, we're mainly working on identifying drugs from plants, natural products. Complete change of direction away from synthetic chemistry in terms of my Aberystwyth work. In terms of my collaboration with Dr. Hina, I've kept my hand in with helping with synthesis. So, just to conclude, We've made over 200 compounds. We've established a structure-activity relationship. We've got selectivity, water-soluble analogs, dual binding inhibitors, and in assays, I haven't shown you additional assays. We've shown functional activity in cells. Compounds are taken up into cells and have activity in cells. We haven't done any whole animal studies yet. And here's the SAR. This is from making our 200 compounds and understanding structure, conformation, properties of substituents. We understand what's important for selectivity, what's important for activity. And here's our model again, our cartoon. This is our 5-AAQ. We've built out into the hydrophobic cavity through the tunnel and into the adenine binding site. Those are our best compounds so far. I'd like to thank all these people who contributed in greater or lesser ways to the PARP program in general, the Part 1 program over 25, 30 years, and particularly into the tankyrases program. Names I'd particularly like to pick out are Katerina Kumpan, Amit Nathubai, and Helen Payne. And I'd like to particularly point out my crystallographic colleague, Larry Lehtiä, who's at the University of Oulu in northern Finland. And these collaborators contributed in their various ways, and a number of visiting scientists and collaborators in the PARP program in general. I'd also like to thank these organizations for providing this very necessary reagent, money. We couldn't have done it without financial support from Worldwide Cancer Research, University of Bath, HEC, Pakistan, and the Medical Research Council in the UK. And finally, I'd like to thank you as well, all of you who have taken the time to turn up and listen, hopefully listen. And I will say that a lot of this work is published, and you can go and read the papers if you want more details, or contact me. Thank you.
Thank you, Mike. Sena, you would like to coordinate the question answer session now?
Yes, sir, sure. Thank you, Professor Mike. Such a wonderful, excellent talk. And I see many questions, but quickly I'll read some of them. Dear sir, what are the activities of these compounds on normal cell lines? This is one question.
That we've not studied very extensively.
Okay.
They do. Their activity is mainly anti-proliferative in terms of cancer cell lines. We've tried a so-called normal cell line, and it seems not to have particularly strong activity. I can't remember the data at the moment.
Right. I think this is related question to the previous one. If any of the compounds showed toxicity against the cancer cell line and normal cell line as well, so what are the possible ways to reduce the toxicity against normal cell lines, and how the cancer cell inhibitors related to the diabetes? I think she is asking about the relationship between cancer inhibitor and anti-diabetic compounds.
It's the anti-diabetic work and the cancer activity are separate. They're both functions of tankyrases. We approached the project from the cancer angle, but then read the literature and found that tankyrases were also involved in diabetes, and started to collaborate with David Tosh in the biochemistry department at Bath, who has an interest in diabetes. So we were not aiming at diabetes, but that was a fortunate outcome, that the same enzyme, same targeted enzyme, is involved in both.
Right, sir. So there is another question and comment. Thank you, sir, for sharing your science and contributing to this event. Would you please share your opinion on the off-target interaction slash binding due to so many nitrogen with respect to the series containing quinoline portion?
We've not studied that extensively. What we have done is looked at NAD requiring enzymes. We should look at others. We should look at a panel of enzymes, I think, and I've recommended that to Dr. Amit. But data I haven't show that, particularly for the lead compounds, for the very potent compounds, which were selective for tankyrases and tankyrases 2 and did not inhibit PARP 1 and PARP 2 well. They also do not inhibit PARP 3, PARP 10, 11, 14, and I can't remember which other PARPs. We tested them against a panel of other PARPs as well, and they do not inhibit those other PARPs. They're very selective for tankerase 1 and tankerase 2. We only used the 1-oxidoreductase, which was IMPDH, actually IMPDH2, and showed we couldn't inhibit it. The enzyme was active, and we could show we could inhibit with a standard inhibitor, but our compounds do not bind to IMPDH at all. And there are papers in the literature from probably 10, 15 years ago saying that PARP inhibitors and don't have crossover inhibition with oxidoreductase inhibitors, although both the compounds bind at the NAD binding site. There's something to do with conformation, something to do with preferred conformation, that the NAD binds in an extended conformation in poly-ADP ribose polymerases, PARPs, but in a curled up conformation in the oxidoreductases.
Okay. Thank you so much, sir. And I see there are some questions which are related to the question which we have just asked, and you answered very well. So with this, I think we can thank Professor Mike for his time and such a wonderful talk. Sir, would you like to add something?
Nothing but to thank Mike that we are eagerly looking forward to have you with us in Pakistan. We really miss you very much and hope to see you soon. You're vaccinated, so please do travel to Pakistan as soon as possible.
Yes, but I will be imprisoned when I come back to UK at the moment.
So you stay with us then longer.
Thank you very much for this opportunity. Thank you very much.
Thank you very much, Mike. Thank you. Thank you very much. Thank you, everyone.
Thank you,
everyone. Recording of this lecture would be available on our YouTube channel, Comstech YouTube channel, and all the participant students will be receiving their certificate for attending this important webinar. Thank you so much. Looking forward to see you again. Thank you, everyone. Bye-bye.
Thank you for the opportunity. Thank you.
Thank you.